Hello everyone, I'm Barrett Nuttall. Today I'm going to be speaking about AZ700, which is an exploratory NGS panel for use in translational oncology. So just to set the stage for AstraZeneca Global Translational Medicine Genomics and who we are, the translational function is in between preclinical and the clinical functions, and we're really trying to translate our results to and from the clinic. So with the preclinical work, we're performing model verification of cell lines of patient xenografts, and then we're trying to take the disease biology that we discover and forward translate it to the clinic. From the clinic, we're verifying that new methodologies and technologies are acceptable and have potential to become companion diagnostics. And we're also performing sequencing on patient trial samples and trying to identify biomarkers of response and resistance. We are primarily a sequencing facility, and we perform DNA-seq and RNA-seq. Today I'm going to be talking about a large DNA-seq panel. We perform a wide variety of DNA sequencing. We perform targeted and exome and whole genome sequencing at quite a clip. So to orient you to what the typical sample is that we're working with, it's usually FFPE tissue material, and the material is often quite limited. As you can see, here's an example of a very small core needle biopsy. There are additional challenges when working with FFPE beyond the limiting sample type. DNA is often quite damaged and less amplifiable. So we really need to identify and deliver robust methods to have extremely sensitive assays. We're most interested in assaying somatic mutations around 1% to 30% variant allele frequency. So just to take a look at our workflow, we are a sample to sequencing lab hosted internally. So we perform nucleic acid isolation and library preparation leveraging some automation. We perform enrichment using twist and competitors, hybrid capture methods, and that's generally performed manually with sample plexing to ensure higher throughput. And then we have a NovaSeq for sequencing, and we have 2 hosted informatic pipelines internally. So for our large pan-cancer panels, we've taken the progression that bigger is better. We've started with a 300 gene panel back in 2015, and now we've graduated to a 700 gene panel just earlier this year. One thing to note is that because sequencing has become more economical over the years, the cost of running this panel has actually decreased while we've been able to increase content. So AZ700 is the first time that we're using twist probes and reagents for this large pan-cancer panel. And this was due to a head-to-head exome comparison that we performed a couple of years ago, where we looked at the performance of the twist core exome versus one of the competitors' exome, and we were able to recapitulate this uniformity plot that twist often demonstrates. We saw that for the exomes where we were trying to achieve 100x coverage, we had nice uniform even coverage to 100x, and then we were also showing a decrease in the long tail of overrepresented reads. So with that in mind, I'll kind of talk a little bit more about the AZ700 panel. It's not actually 700 genes exactly, it's 697, but AZ697 just doesn't roll off the tongue quite as nicely. It has 1x tiling across all the total regions of interest, which are all, it's just exonic coverage. And then we have 2x denser tiling in critical genes and difficult to capture regions. So the critical genes are the cancer hallmark genes where we need to ensure that we have high coverage. And those difficult to capture regions are areas that we discovered had poor coverage from previous AZ650 sequencing and also sequencing with the twist core exome. So just to demonstrate the effect of this 2x tiling, Here is a cohort of FFPE tissue sequenced with AZ650, the previous panel, and you can see while we achieved a overall panel coverage of over 1000x, the coverage for the PTEN gene in particular was less than 500x. For a similar FFPE tissue cohort sequenced with the new AZ700 panel, you can see that our overall panel coverage is higher, and that the coverage for the PTEN gene in particular is much closer to the average coverage, and we're getting nice uniform coverage across this gene. So in addition to the regular tiling, there is an optional spike-in consisting of intronic probes targeting gene fusions, and there also are the HLA class 1 genes covered. So to talk a little bit more about the validation process, we had a multi-site wet lab protocol development and validation. It was performed in Waltham, in Gaithersburg, and in Cambridge, UK. We went with a compact design where we had a sample set consisting of 8 samples, and we had a total of 8 replicates of this sample set. The material we used was FFPE-derived cell line DNA with a clinically relevant input of 100 nanograms per replicate. While FFPE-derived cell line DNA is not a perfect substitute and step-in for FFPE-derived tissue DNA, we felt it was appropriate, and we were able to identify some excellent reference standards for use here. So we aim to deliver the limit of detection, precision reproducibility metrics, sensitivity for structural variants, evenness of coverage, identify what the achievable depth for qualified samples are, and then we also performed an HLA benchmarking experiment for this validation. I'm not going to go into the outputs of all the deliverables, but I'll briefly talk about the limit of detection and the reproducibility metrics. So for the limit of detection, we went with a streamlined, more stringent definition. And for AZ700, that is the variant allele fraction at which all replicates for a variant type are detected. So for AZ700, our limit of detection came out to be 0.9%, and for SNPs and small indels. So you can see we generated an output similar to the MSK impact limit of detection table, and you can see at 0.89% is where we were able to robustly call all 4 replicates. So we also created the precision and reproducibility metrics. So out of the 8 samples, we considered 5 a subset, and we were analyzing 270 variants per each of those 5 samples. We had known presence and absence of each of those mutations. They had either been previously sequenced internally, or we were able to identify from public sources or from commercially procured sources the mutations that were present or absent in these samples. So in that way, we were able to define the known true positives and true negatives, and we also included within this 270 truth set known artifactual calls that we knew would occur, which could be possible false positives. So we returned our precision metrics in 2 ways, using a relaxed set of filters where variants only had to pass a very lenient filter to be called, and these relaxed parameters will allow for false positives. And then we used a stringent calling filter set, which we know will remove artifacts and remove mutations of unknown significance and only return mutations of known and likely significance. So here is just the output. This is the kind of raw data from a relaxed filter set, and you can see the 5 samples that we were using to call here. And here's the site-by-site and overall metrics for the relaxed and the stringent filters. So you can see with the relaxed filters, because we had false positives coming through, our specificity was not 100%, but it was still quite good. When we apply the stringent filters, all false positives and artifacts are removed. We had 100% sensitivity, specificity, PPV, and NPV down to 1% variant allele frequency. So pivoting away from the validation and to talk a little bit more about the production informatics and why the alignment algorithms and their settings matter, when we initially processed the data with our internal BC Bio pipeline, which uses BWA MEM aligner, we were seeing that about 8% of our area region of interest was receiving no coverage. And here's an inset highlighting that. When we dug into these areas with low or no coverage, we discovered that the majority of them were alternate alleles, alternate contigs. They were receiving almost no coverage. We then performed processing with the Dragon box using it uses the drag map aligner, and we were able to see that we could rescue coverage in all these alternate alleles, and we had nice even high coverage out to 1000x. And just to highlight the difference here, we were seeing that we rescued the coverage in the alternate contigs. So the lesson learned is that if you're seeing a drop in your performance metrics like this, it may not be due to your probes, and it may not be due to your laboratory process. It might be due to your aligner, and that's something to keep in mind. So BWA MEM can also be tuned so that you are able to better align to the alternate context. So keep that in mind. BWA MEM is not a bad aligner, but aligning to alternate contexts may not be for everyone. If you are going to be performing HLA calling, you will need to enable this kind of alternate contig alignment. So kind of to go into a case study here. So FFPE breast tissue is a notoriously difficult sample type to work with. Breast tissue often has low cellularity due to adipose tissue. So your actual nuclear content, as opposed to surface area, is relatively quite low. So we had a set of 200 triple negative breast cancer FFPE tissues, and this is from the AstraZeneca TICA, Tumor Immune Cell Atlas cohort. And TICA is you can think of it kind of like AZ's internal mini TCGA, where we're trying to layer multi-omics levels of data on top of each other to better understand the tumor microenvironment. So we had originally processed the sample set in 2016, and the exome data quality made it difficult to call somatic mutations. The average coverage was lower than desired. It was about 200x over the entire cohort, but the uniformity was quite low, and our critical biomarker genes generally had lower than 50x coverage. So the samples were reprocessed with exome sequencing again in 2020 under using two different parameters, and we saw that the coverage and overall results were worse. We went back and looked at the average DNA quality ratio. This is a metric from the kappa HG quant kit, and we saw that over time from 2016 to 2020, the DNA had slowly been degrading and becoming less and less amplifiable, which explains why we saw worse results with the exome. So this sample set was kind of ripe for reprocessing. We had additional tissue left over, and we really wanted to be able to generate high quality data. So we performed the whole sample process of extraction to library preparation one more time, but this time instead of performing whole exome sequencing, we used the AZ700 panel. So just to give some relative metrics, the AZ700 validation samples, which are again cell line FFPE, are shown here, as well as the TNBC cohort samples. And you can see for the TNBC FFP samples, the DNA quality ratios, the median was about 0.3. The quality ratios are still quite low. So while we're good, we're not magic. We couldn't make the DNA more amplifiable and better quality than it was previously extracted at. But we saw that when we went to sequencing, the percent on target was actually higher than what we achieved during our AZ700 validation. And this was because we identified some process improvements in our heating for our wash buffers. So digging in a little bit deeper to the QC metrics, we saw that the post-UMI consensus coverage was over 1500x for the TNBC cohort. And while the AZ700 validation sample set did overall achieve a higher coverage at just over 2100x in total average, we were very pleased with this result from the TNBC cohort. So we not only achieved high coverage, but we also achieved uniform coverage. So this is displaying the fraction of AZ700 with greater than 500x coverage. And for the TNBC cohort, it's at about 88.5%. So we are getting greater than 500x on the vast majority of the content that we were trying to capture. So this is overall a great success story because we were able to salvage a really robust sample set and bring it in, and now we can start integrating these multimodal omics methods and combine them all together. So just to give a peek on how you would actually start to perform this interpretation work, we're really looking to identify the prevalence of mutations and the co-occurrence of mutations. So this is again the TICA TNBC cohort data, and these are the same samples. And this is just a visual representation of the relaxed filters versus the stringent filters. And you can see when you apply the stringent filters, you remove a lot of the noise, low quality variants, and you can really start to interpret the results and the patterns within your data. So I just want to say thank you to everyone, the AstraZeneca team and the Twist team. It was an amazing effort by many people. And I hope you all are now inspired and motivated to go create your own large gene panels. I think with how economical sequencing has become, we will soon see the prevalence of 1,000 or larger gene panels being able to call down to 1% variant allele frequency. And with that, I'd just like to say thank you to everyone. Have a great day.